before we start
Yeah.
the webinar. I have the request to record the meeting. I don't know if you have some problems with that.
No,
It's
I'm
okay
fine.
with you? Okay, perfect.
I cannot share. I need to be habilitated.
Okay. All panelists, okay. Panelists, okay. You should be already
Yeah.
habilitated. Okay, perfect. Um,
I
I
think.
only need support for the platform. I will be mute and no video for me. And I will be here. And anything. I will start the
Thank you.
webinar in this moment. Okay, welcome.
Okay. Okay. So how is gonna be? Francisco, you're
Yeah,
online, yeah?
I'm going to introduce the conferences,
Mm-hmm.
and
Are
once
we waiting a bit for people to connect?
we're opening the talk right now, so we're gonna see. Okay,
The
so we
webinar
already.
is already started. We have 67 participants. We're going 70 right now, and the number is great. Um,
Okay, perfect.
let's keep 1 minute to start.
Okay,
Okay.
so I'll just let you know, Damiano. I'm going to introduce you in Spanish for the students,
Yeah.
and then I will introduce the seminars, introduce you, and then I'll give the talk back to you. Okay. At the end, when the students make questions, I think they're gonna have to write them down. If they write them down in Spanish, I'll read them and translate them to you. Okay.
Okay.
By the way, it's great to
Recording
see
in
you again.
progress.
How is the view? So do you see the slides?
Yes. Yeah. Yeah. Molecular
Uh,
dynamics methods to study and
what
defend.
about what about like this?
I still see them, and I can see your mouse pointer.
Okay. Like this? There's a pointer.
Yes.
Mm.
Muy buenos días a todas las personas que están conectadas, a los estudiantes, profesores y compañeros que están conectados. En unos momentos vamos a iniciar el seminario. Este, de parte de la Universidad de la UABC, de la Facultad de Ciencias Químicas e Ingeniería, les agradecemos mucho su asistencia. Y en unos momentos comenzamos. Okay, bueno, ya son las 8:10. So, Damian, I'm going to start the introduction. I'll start in Spanish and then I'll get back to you. Eh, nuevamente, muy buenos días. La Universidad Autónoma de Baja California, a través de su Facultad de Ciencias Químicas e Ingeniería, les da las gracias por participar en este evento. Estos son unos ciclos que se están realizando de conferencias de investigación, divulgación científica e industria por parte de la Coordinación de Investigación y Posgrado y la Coordinación de Extensión y Vinculación de nuestra facultad. La intención de estos cursos es proporcionar un amplio panorama para los estudiantes para que puedan conocer diferentes áreas, tanto de investigación del área científica o de la industria, y ellos puedan ampliar su panorama, conocer las oportunidades que tienen y puedan tener un mejor desarrollo profesional. El día de hoy nos da mucho gusto poder presentar al doctor Damiano Buratto, profesor Damiano Buratto, quien se encuentra trabajando actualmente en el laboratorio del profesor Michale Levitt, quien el doctor Levitt ganó o fue galardonado con el premio Nobel de Química en el 2013, justo por el desarrollo de metodologías de dinámica molecular de ciencia computacional. El doctor Buratto es de licenciatura, es físico, se graduó en la Universidad de Padova, Italia, y posteriormente estudió su maestría y su doctorado dentro de la misma universidad. De ahí se movió a hacer una estancia postdoctoral en la Universidad de ShanghaiTech, en el Instituto de Shanghai Insitute for Advanced Immunochemical Studies, conocido como SIAIS, en el laboratorio del doctor Richard Lerner, quien fue director del Instituto Scripps de aquí de San Diego, y el trabajo del doctor Buratto empieza a tornar de las ciencias computacionales aplicadas al área biológica, dinámica molecular, pero ahora con anticuerpos, debido a que el laboratorio del doctor Lerner se dedicaba al desarrollo de anticuerpos monoclonales. Dicho sea de paso, el doctor Lerner desarrolló el primer medicamento a base de un anticuerpo monoclonal, que es conocido como adalimumab, o la patente era Humira, que habría sido hincapié que el doctor Lerner ganó alrededor de $8 millones por su participación y desarrollo de este medicamento. Entonces, la intención es que ustedes conozcan la parte de computacional aplicada al área biológica, y que tengan en mente que si uno sabe explotar adecuadamente las metodologías de investigación y las técnicas de laboratorio, uno puede desarrollar ciencia aplicada que puede patentar y que puede generar un ingreso. Entonces, el trabajo del doctor Damiano Buratto el día de hoy se titula Molecular Dynamics Methods to Study and Design High-Affinity Antibodies. La presentación del doctor Buratto va a ser en inglés. Cuando tengan preguntas las pueden ir escribiendo en el chat, y al final, cuando sea la sesión de preguntas, las vamos a estar se las vamos a decir al investigador. So, Damiano, again, thank you very much for joining us. It's an honor for us to be able to have you as a panelist, and now I will you can start your talk now, and we can see perfectly your slides.
Okay, thanks. Thank you very much for the kind introduction, and yeah, for inviting me for this seminar. I hope you will enjoy it. So in this seminar, what I'm going to do is to have a brief explanation of what you can do with molecular dynamics, and I will show you some methods that we used to study and to have designed some new antibodies that has a quite good high affinity with their target. So first, I want to start with, what is actually this molecular dynamics? So molecular dynamics, as a definition, is a computational simulation for analyzing physical methods and atoms. This methodology can be used on different fields, like chemical physics, where you can use it to guess chemical transition. In material science, they can use it to study thin layers of material, and in biophysics, where we use it to study the dynamics of macromolecules. Of course, on this seminar, I'm going to talk about the biophysics part, and how is actually working this molecular dynamics. Well, what you need to run the molecular dynamics is a potential function. We call it force field. This is a description on how every particle will interact with each other during the simulation. There are many different molecular dynamics that you can use, and everyone depends on the specific problem that you want to address. In this case, we are going to see all atom molecular dynamics. As the title says, all atom, so this kind of molecular dynamics require you to have the coordinates of every single atom of the system that you want to simulate. And the force field that usually are used here are mostly empirical force field, and they consist on a summation of bounded forces and non-bounded forces. So the first thing that you need is a huge file with hundreds of thousands of atom numbers and name, and residues number and name, and the spatial coordinate, the X, Y, and Z for every single atom. If you feed this file to Bright software, you can have this very fancy picture where they show you actually how is your system. In this case, we have a small box of water. The second file that you need is the topology file. In this file, you have all the atom properties, all the peptide bonds properties, all the amino acid properties, and there are parameters that like the charge, the radius of the atom, and other parameters that the program needs to define the force that will interacts, the force that is used on each atom. So using both of these files, you can start your algorithm. There are different software to run molecular dynamics, and they generally all follow the same iteration. They start with the initial position that you set, and they compute the force for every single atom. And with that force, they predict how every atom will move and where. After the prediction, there are some adjustments that depends on kind of simulation that you are doing. After the adjustment, they just restart and the process go on. What you have is this kind of simulation. Every atom starts to move in a very meaningful way. If you see that there are some atom that is flickering, that's because there are some boundary condition, and that means that when this atom go toward the bound, the box that we define, it will appear on the other side. That's a small trick that we use because we cannot simulate an infinite number of atoms. Okay, well, how and what do we use the molecular dynamics for? Usually in biophysics, we apply molecular dynamics to study the motion of the macromolecules. And well, this helps to interpret the results and gives you the possibility to have predictions. Well, it's important, first of all, to have an idea of the dimension of what we are doing. For an average full atom system, we have around 200,000 atoms, and the box is a cube, or generally a kind of cube, and every dimension of this cube is generally below 12 nanometers. So it's very small. It's like a point, just a small point on the surface of a cell. And of course, more atom you have, more the software needs to work on it. So with a system with this number of atoms, you can simulate with a good computer around 20 nanoseconds a day. That's nanoseconds. And you generally goes around a good simulation, you may want to arrive around 100 of nanoseconds. So that's what we are talking about. It's very, very small time step and in a very small environment. What can we do with this? Well, we will see how we use it for have a better understanding of the molecular mechanism, how the antibody can dock the protein. We have prediction on how the antibody can dock different proteins or how we can stop the antibody to work. And then in the end, we can also modify protein and design new antibodies, for example, for specific purpose. First of all, I want to be sure that we have everything clear of what we are talking about. So we start from introducing everything. So proteins. Proteins are large biomolecules, and these are like made of a chain of amino acid. Here we have the 20 standard amino acid. That's all the simulation are done with these standard amino acids. Every amino acid shares the same topology, let's say, the same topology. Well, the same, yeah,
Backbone?
and what is different for every amino acid? Sorry? Yeah, backbone. Yes. Yeah, I didn't want to use backbone, but yeah. Every amino acid, the difference between the amino acid is the side chain. The side chain is highlighted with the color in this image. So different side chain gives the amino acid different properties. Every amino acid can dock together and form chain of amino acid. The bond between two amino acids is called peptide bond. And so here we have the first level of the protein is the primary structure, and that is a chain of amino acid. So basically a list of amino acid that will define your protein. That in general, let's say, is not sufficient to know exactly what is your protein or what it's gonna do. And these amino acids start in nature will fold and form different structure. The two most common structure are this secondary structure that are the beta sheet and the alpha helix. And you will find, so in the 3D, actual 3D structure of the protein that is called the tertiary structure, you will find a lot of this beta sheet and alpha helix. So the 3D structure actually is what will tell you how is the shape of the protein. But many proteins will work together to make different agglomerates like channels or other macromolecules. So different proteins that dock together are called the quaternary structure. We will see mostly many proteins that will interact together to work. After that, well, it's clear that we are going to talk about antibodies. So what is an antibody? An antibody are proteins of the immune system, and these are used to recognize and neutralize the foreigner objects, such like bacteria and viruses that are so infamous in these days. The typical shape of an antibody is a Y shape, how you can see here. And why antibody? Well, antibody are a very important family of protein because they can bind with a very high affinity and very high specificity to their target. What does it mean? It means that they bind very strongly and exactly to the target that you are pointing and not to something else. That's why we started to develop new antibody in the lab, and we developed them to bind specific targets, like kill some other harmful macromolecules or bacteria or other stuff, or we use it to dock to other proteins and eventually activated them or deactivated them. And they are also engineerized with fluorescence labels and used in research, for example. What is important in the antibody? So apart from the Y shape, the most important part of the antibody, for our purpose at least, is the complementary determining region, CDR for short. And my connection is unstable. Seems okay. So yeah. So what is this CDR? Basically, this CDR is the most variable part of the antibody. So it's the part that is mostly changed between different antibody, and it's what allows the antibody to dock to the exactly target that you want. So now we need to keep always in mind that with larger system you have slower simulation. So this is our typical antibody, but we are going to use only this part. This is only one arm of the antibody with the CDRs. And many times you will actually see only the CDRs, because this is the smallest part that we are interested in, and that is the way that we use, the trick that we use to speed up the simulations. So now I want to introduce, well, what mostly what I've been working with. These are connexin protein. Connexin is a family of proteins. So there are many proteins that share the same shape, let's say, the same topology. And these connexin form channels, gap junction channels. So we have six connexin docked together in a hexamer shape, and they form a hemichannel that goes inside a membrane of one cell. If this hemichannel is able to dock with another hemichannel from the adjacent cell, then they are able to form a full gap junction channel. This full gap junction channel is very important. You can find it virtually in every cell of your body with different connexins, and they allow the transfer of material and information between adjacent cells. It was like 2009 when they managed to do the first crystal structure of the connexin 26, that is one specific connexin of the family. Thanks to this crystal structure in my group, we did the model of the full gap junction channel, and in a realistic environment with the membrane and the solvation with the water to solvate and the ions. Here on the bottom left, you can see the topology of every connexin. So they have 4 transmembrane helixes, so these alpha helix that are inside the membrane, and on the top side, on the top part, so outside the cell, we have 2 extracellular loop. For my study, what I'm gonna show you will always use a hemichannel. Why hemichannel? Well, not all the hemichannel are able to dock with another hemichannel on the adjacent cell. And this hemichannel, most of the time, should be closed. Unfortunately, there are some mutations that generate functional hemichannel that are leaking. So if you actually have this kind of mutation, you have very bad disease, and these are due to the fact that hemichannels are leaking material and the cell is losing material from inside. So in the project that we are working in my group is about an antibody that can sit on top of this hemichannel. The idea was like just go inside the pore and plug it. So we want to have an antibody that can rescue this mutation and stop the leaking hemichannel from leaking more material. Well, I will skip all the experimental part. They managed to fish one antibody that actually is able to dock the hemichannel, and one kind of measurement that we can do to see if the antibody is working, we can measure the current that passes through the hemichannel. So if the antibody is working, we see that the current is damped. So we have almost zero current when we use the antibody. After the development, after they find this antibody, we started to build the molecular model. So we build an atomistic model of the antibody. We place it on the top of the hemichannel, and for how it was developed, the idea was that it should have go in the middle and just block the hemichannel. What we actually saw during the simulation, is that it start to move and it start to go on one side of this hemichannel. This, well, was not expected, but we thought about it, and probably that means that we need two antibodies on the top of the hemichannel. So how we can see, this is the single antibody, and it can fit a second antibody perfectly like this. After we build this new model and we run molecular dynamics, the dynamics, it was stable, so it was actually a good model. And with further experiment, they actually confirmed that the stoichiometry of this interaction is 2 to 1. So we have two antibodies for every hemichannel. So this was one prediction that we managed to do with molecular dynamics. The second thing that we want to do here is to confirm that our model is actually able to reproduce the experimental results. So what we have in the experimental part is we measure the current through the hemichannel, as I said before. So we set up a couple of molecular dynamics simulations, one without the antibody and one with the antibody, and we set a voltage, so a potential that make easier for the ions to pass through the hemichannel. And what we saw, is that without the antibody, the ions can actually go through the hemichannel, and we can measure a good current. With the antibody on top, clearly the ions has harder time to pass, and the current that we managed to get from this simulation are these two, and how you can see with the antibody is much damped, is much lower compared to the one without an antibody, and the ratio between the two currents is has a good is very close to the experimental results that we had. So this was a quite good achievement to say that our model is actually working. And so we wanted to go further with the predictions. So we analyzed the molecular dynamics that we did with the two antibodies on the top of the hemichannel. And the analysis that we did is we wanted to study which residues of we started from the antibody, which residues of the antibody is responsible for the docking. So we want to understand which of the residues is mostly important in the binding. So on these tables that we get from the dynamics, we have a percentage of interaction of different residues of the antibody, and we did a visual inspection knowing this table, and we saw that these three amino acids are having a very strong interaction with the hemichannel. We suggested these three amino acids to the lab that they did single mutation on these three amino acids, like so they changed the residues like the 104 and then 55, then the 34, with a residue that is completely different. And the result is that if you modify any one of these three, the antibody is no more able to dock consistently with the hemichannel. We did the same thing, but on the other side, on the hemichannel, and we want to understand which residues of the hemichannel are mostly important. So how you can see here on the right, the hemichannel, the two antibodies dock exactly in a specular way. One antibody takes the orange part, and one antibody the blue part. And the residues that are mostly important, following the same procedure that we did before, are these that are highlighted by the red squares. What can we do with this information? So as I said, connexins are a family of protein. All this work has been done with connexin 26, but there are many other connexins in this family. And we want to understand if this antibody can actually dock to other connexin of this family and how strongly they can bind. And we want to have a prediction of that with this data. And to do that, what we did is a sequence alignment of the connexins. What does it mean? It means that we took all the amino acids that form each connexin of the family, and we put one next to the other in a very ordered way. And what we did is an analysis of the residues that I showed you before that are, for us, are the most important for the docking. And so this is connexin 26, HCX26. And here we see the residues I showed before. And we can see that the same residues are in the same position for connexin 30 and connexin 32. The other connexin of the family, instead, they always have at least one mutation on this part of the connexin. So with this result, we said that most probably this antibody will be able to bind also connexin 30 and connexin 32, and we'll have trouble on binding on other connexin of the family. And we actually were right. They did the experiments, and as you can see, these are the results on the current. You have 100%, and here you have the current for connexin 26, while connexin 26, connexin 32, connexin 30 with the antibody, and the same antibody with other connexin has almost no interaction. Okay, so for now we have done for the connexin, and the last part I want to talk about the design of a new antibody. So the maturation of an antibody is a quite well established method from the experimental point of view. So they start from an antibody that can dock quite well with the target, and they introduce mutations on the CDRs, so on the part that interact with the target, hoping to increase the affinity, so to increase the binding with that target. The idea that we had is to reproduce this methodology, but doing that in silico. That means with the computer. So I started to develop the software to do this work, and this software starts with a starting configuration like this one that we have, the antibody. In this case, like only the one arm of the antibody with its target, and we select one residue on the CDRs. We mutate that one, so we change it for a different one, and then we run molecular dynamics, and we compute from this molecular dynamics this binding energy. What is this binding energy? Well, this binding energy is a measure of that we can have from the molecular dynamics, and it's well correlated with the experimental binding energy. So it's basically the correspondence of how strong the antibody can bind to that target, and it seems to work quite well. It has a very good correlation with the experimental value, so it can be used for this purpose. So this is the whole program. So if the binding energy of the new configuration is better, then we keep the new configuration. We go back and we modify a new residue. If it's worse, we go back to the previous residue and we start again. I tried to apply this one for one configuration, starting from an antibody that we had and was well studied already in our lab. So it was like a test model. And well, it was already quite good, actually. It has nanomolar affinity that's the binding was already pretty strong. And I ran this software 3 times, having 3 different results. And 2 out of 3 had a binding affinity that has been tested experimentally. These are experimental results, and is actually 3 times better than the one from which I started. So this is one way that we use to design a new antibody that is actually binding stronger than the starting one. And with this, I finish my presentation. I want to thank members of my group, like my professor, Michale Levitt, Francesco Zonta, and from University of Padova, my previous supervisor, Fabio Mammano, and other co-worker there. And especially want to thank the University Autónoma of Baja, California for inviting me, and Francisco that invited me for this talk. And I hope you enjoy the talk, and I'm here if you have any question.
Okay, Damiano. First of all, let me thank you very, very much for your amazing talk. Although I had taken many coffees with you, talked about a lot about science, I had not actually seen how you developed the antibodies. And I remember the experiments you had done with the connexins. So it was great to hear how you did the experiments. And I'll get back to you in 1 second. Ahorita vamos a empezar la sesión de preguntas. Entonces, si les interesa hacer una pregunta, pueden escribirla en el chat y yo la pueden escribir en español o en inglés como gusten y yo le haré la pregunta al doctor. Entonces, piensen las preguntas, le voy a hacer yo tengo unas preguntas a él de momento en lo que empiezan a pensar en preguntas. Todos están invitados a hacer cualquier tipo de pregunta. So, Damiano, one of the things that I remember a lot from Francesco when we were over there, he told me once that you had run an MD simulation, but the antibody, instead of both antibodies sticking to the connexin in this position, they were like in an inverted position, right?
Maybe.
Okay, and also, for example, experimentally, have you guys made sure that you have 2 antibodies on top of the connexin?
Yes.
How
Because so we compute the dose response curve for the current of the hemi channel with different dosage of the antibody, and this curve has to fit that curve, you need to use a stoichiometry of 2. So you need 2 antibodies for every hemi channel.
Okay. All right. The antibodies that you're currently using, are they recombinant ones, or are you still are you generating by recombination and you purify them, or are you purifying them from directly from the mouse and making them monoclonal?
No, actually, this one was found with the library, the phage library.
So it's already you already have the sequence and everything, and you're just making it recombinant. Oh, okay, super cool. Okay, so we have now some additional questions from the attendees, and the first question is from Emiliano Salinas, and he's asking which method did you use to compute the binding energies in the molecular dynamic simulations?
Yeah, we tested different methods. We wanted to be fast and precise as much as we can. Let me see. Well, yeah. So for this result, we use MMPBSA, right? Yes, MMPBSA. Yeah, and this allows us to have a decent speed with the molecular dynamics. This is mostly the first start of the project, and other option was to use FEP. Like, I'm not sure how familiar you are with that, but so MMPBSA is like the average solution, let's say. FEP requires a lot of time to do the simulations, but it's much more precise. For now, the idea was to separate, to see how much we can separate bad antibodies from good antibodies.
Okay, perfect. So now, Dr. Arturo Stolano is asking, well, first he's thanking you again for that very interesting presentation. And then his question is, where did you produce the mutations on the antibody to see the residues more responsible for the interaction with the hemi channel? How did you select these 3 residues you eventually mutated?
Yes.
And
yes, yeah, go ahead.
He says that I see other residues on the table that have high interaction probability. For example, threonine 30 with 100% probability.
Yes. Well, this table is not enough to see if the interaction is actually strong. We combine the table with actually a visual. We watched our model on what is actually happening, and these 3 residues happen to be inside pockets or inside or in interaction, having strong interaction with other residues, like making hydrogen bonds, or where one was exactly I don't remember which one. One it was exactly in the middle of a fork, so it was in a very strong position. The other are still always interacting, but you need to select, visualizing actually what is happening.
Okay, perfect. Okay, so there's another question from one of the students, and she's asking, how's the correlation between the thermodynamics and the studies that you're doing? I think she's mentioning this from the initial part where you were talking about when you had the protein in the box. So how could you explain to us where's the thermodynamics in this?
Again, the thermodynamic. Well, so I'm not sure. Well, I can just. So the thing is, during the simulation, you have different. So apart. Okay, let me go back. Here we go. So apart from computing the force like with brute force and with approximations, there are a lot of approximation during these simulations, and that makes necessary to introduce some correction. For example, depending on the simulation that you are running, you may want to have a different environment, like NPT or MVT. So like a number of atoms that are always constant, pressure can be constant, and the temperature can be constant. Or you can have temperature constant and volume that is constant. So you can run different things, and you have some kind of helper, let's say, that will correct the simulation for keeping the temperature constant or the pressure constant. I'm not sure if that answered.
Okay, more or less. So I guess what you do is like you take into account all of the like the structure and how's the position of everything, and you're able to calculate how much energy is in the system, no? And when you're doing the dynamics and the molecule is moving, you're able to continue calculating everything and seeing, I don't know, the free energy of Gibbs or something, no? You're checking that all the time, no? And the entropy and on all of the parameters, and you always know like the energy that the system has.
Well, the energy depends on the temperature of the system and the speed that every atom has. So the speed will give you the temperature of your ensemble.
For example, I remember when we were collaborating, and you were helping me out with the structure of a protein that we determined. I would give you the model, and I remember you were able to minimize the energy, right? Relax the structure and everything. How could you explain to us how exactly you do this, and what's the fundamental of relaxing the protein?
Yeah, that's because that depends on the force field. So the force field is a set of parameters, like the Coulomb. So for electrical, the charge of the atoms, the van der Waals that defines more or less the shape, the surface of the atom, and then you have other bond interaction, angle interaction, dihedral interaction, and all these interaction has parameters set on the force field, and these are used like to compute the force from a potential. So every atom has a potential. This potential is energy, basically. So from there you can compute the energy of the binding or the energy of the molecule, or whatever you need. So, I don't know.
Perfect. Great answer. Another student, Fidel Serrano, is asking if you can make an antibody with the same binding affinity with less amino acids. I mean, like if you can actually make the amino acid, the antibody shorter and still have it possess the high affinity.
It's a possibility, but we didn't try. We were not. We didn't try that. It's more difficult it requires more coding, I guess. Still doable. We just didn't try.
Okay. And just before I make the next question from one of the students, just for the students to have an idea, what programming languages do you usually use to do all of this modeling? And for example, there are
many languages that you can use. Python for sure is one, and inside Python there is OpenMM, that is a software to run dynamics. Otherwise, there are other software like Ember, but it's commercial. Other free software like Gromax to run dynamics. For visualizing, there is one of the most famous like Chimera or PyMol, VMD. These are mostly the most famous software for the visualization. Well, Schrödinger, of course, is very famous, but commercial. For coding, you may need Bash a lot, mostly to use the high-performance computers and to set scripts. And then depends on which software you are going to use. They have different languages, but I mean they are generally all the same, just small keywords of difference.
would you suggest that the best option is Python or C++?
Depends on what you want. If you want a fast program, is C++. If you want a very good program, like Python can do everything, and you can really use it for everything. You can connect it with everything. Python is amazing. C++ is a little bit faster.
Okay, one of the other students asks, what do you enjoy the most about what you do?
Publishing the paper, probably.
I would have thought, I would have thought you would have said having a coffee while my simulation is running with my buddies from the institute, you know?
No, like be able to do right prediction is really good. Like I'm very satisfied for that. So when you manage to do something that is actually working and you can prove that it worked, yeah, that's really good. And in general, I like the environment, or the work environment. I like to work with my colleagues, and I enjoy mostly that, I guess.
Yeah, I think that's one of the most amazing parts, no? And discussing science
Yep.
while you're having a coffee.
Sugar with coffee.
Yeah. And so now, one of the other attendees, Summer, asks, well, she first starts with, great talk. Could you explain in your model how the two docked antibodies blocked the leak CX? Is it physically blocked or induced CX conformation, a conformational change leading to a smaller leak?
So it's a little bit of both, actually. We saw that there is a conformational change, and the pore seems to be a little bit closer, probably because the two antibodies dock and then they dock with each other, so they push the AMI channel. But in general, I would say that most of the work is done by the two antibodies that are actually clogging the AMI channel.
Perfect. Excellent question also, by the way. Okay, and Lisbeth Molina is making the question, what interests you most about this specific area that you chose?
So this is quite like we are able to apply this quite like relatively fast, let's say. The idea was actually to develop this antibody to have a cure for this disease. These are very rare diseases that generally nobody cares, but they're quite nasty. And it's good if we manage to have something to help this.
The patients. Great answer. Very noble answer also. I hope more researchers would follow this, and pharmaceuticals would also try to do this to help people. So this is an amazing question, that we have right now from Alejandro Carballo. His question is, what's the present or future on MD or molecular dynamic antibodies with the potential tools such as AlphaFold?
Yeah, well, the hope is that we are gonna send all the experimental guys home in retirement,
Come on.
and we can just do simulations.
Perfect.
Well, AlphaFold did a great step because it almost solved one of the biggest problems in biology, like starting from just the sequence, they managed to have the 3D structure of a protein. And that was a huge problem for many years. And the results was just astonishing. So
I would like to ask you for next semester if you could help us with we talk about AlphaFold, to open our minds about what are all of the uses of this tool and how it works, Um, Another of the attendees asks, Raúl Celis is asking, what are molecular dynamics trajectories?
Sorry, I didn't hear you.
What are molecular dynamics trajectories?
Oh, well, so clearly you cannot have the position of every atom for every frame of your trajectory because the file will be huge. So generally they are binary files, and they are read from the software that you use to visualize in it. You need you always need, so the trajectory alone doesn't work. You always need the initial coordinates and the parameters for every atom. And that's a way that in this way, trajectory are a little bit less heavy, but still you get gigabytes of trajectory.
Okay, nice, nice. Another of the students, Kevin Enrique, is asking, what's like the main reason why you chose to study this? And I think it's a little bit linked with the previous question that you answered. That was what you liked about the research that you're doing, no?
Mm-hmm. Yes, yes. Yeah, and I also enjoy like this molecular dynamics working with computer. I always liked the programming and these things. So yeah, I guess I started for that, and then I enjoyed this field more and more.
Perfect. Uh, Yara Jasmine Lopez is asking, can you use another cellular connection for this project, or can you only use the gap ones? Like, she means like the proteins that are for cellular connection. Are you only going to be working with this connection, or are you planning to work with additional ones?
With different proteins, you mean? Yeah, of course. The idea would be also to work with different proteins. These methods can be used in general. They are not specific for this target. I work on this target, but in the future I may change group, may work with different things.
Hopefully, in the near future, you will start your own group. I hope for that. And again, Raúl Celis is asking, and can we have a different curvature in different types of antibodies? For example, if we were using an IgA or an IgM?
What do you mean? I didn't hear the first part.
Okay. Can you have a different curvature in different types of antibodies?
Different curvature?
Yeah, I guess it's like the binding area. So if the antibody is an IgA or an IgM, so that I think like the area that binds is going to be quite different because the whole structure of the antibody is going to be different, no? So can you apply all of this molecular dynamics to for any kind of antibody?
Yes. These are just proteins. So all proteins are standard. You can usually as a standard in the molecular dynamics, as long as they are protein. If you can also work with other things like small molecules or DNA. It's just a little bit less standard, let's say, but it's still doable.
Perfect. Andrea is asking, how long did it take you to calculate the binding energies in molecular dynamic simulations?
So I think it was like 6 hours for everyone. So like 4 antibodies a day, 4 different antibodies a day, and yeah, the three software run in parallel. It takes time. It takes a lot of time. It around probably 10 to 15 days to complete the whole cycles.
Perfect. Days, so it takes days to calculate this. Okay.
So 6 hours to compute one single binding energy, but the idea is to iterate the program for many antibodies and see many different binding energy. The software, for how we design it was kind of at a plateau around 50 iterations, and so around 10 to 15 days to run the whole program.
Okay, perfect. So actually, you need. This is just like a small idea or a small question, but what are the characteristics of the cluster that you use at ShanghaiTech or SIAIS to run this molecular dynamics? I'm asking this because it'd be interesting if maybe here at our university, because we have a science, computational science department, maybe this could be something useful for us for the future.
Well, we have so what I'm using are we have a smaller cluster for SIAIS. It has like 12 nodes with the same architectures, and these architectures should be 44 CPU and 4 GPUs. I don't remember which GPU though. Then there are other 2 nodes with 33 CPU and 2 GPUs, but these are the newest GPUs, so they are actually better.
Perfect, perfect. I hope we can get some of those clusters here at UABC. So also, Kevin Javier made an interesting question: is could you plan to use these antibodies to mark tumors in the future to prevent them from exchanging ions and possibly to reduce the pathogenic process of cancer cells?
Well, antibodies, yeah, they can be used against cancer, but well, I mean, there are already some working. We didn't have this project, but certainly can be done, I guess. Yeah.
Perfect. And Jesus Gonzalez asks, what has been the biggest challenge that you've been through during your process of doing research in the molecular dynamics field?
Well, I mean, these are the good results, but there are tons of bad results behind this. I guess that's the biggest challenge, because you fail many times and you get one good result. So you need to be still strong and go on, and you will learn from trying and error, because I mean, new things nobody tried. Well, if someone already did it, then it's okay. You can still not that easy, because they are always like the edge of the knowledge. But yeah, I think that's the most difficult thing.
Okay. And Dale asks, what other projects are you interested for the future?
Well, there are different. We have different ideas. One possibility is one project that we are discussing about is the development and to be able to change the antibody that we already have, that's a dock on the connection 26, 30, and 32, and change its specificity to be able to dock connexin 40, connexin 42. These are connexin that are quite different, and they are extremely important for other diseases.
Perfect, because where you stopped in that question, the next one actually starts. Do you know what the pathologies for these hemic channels are? And if you could share that with us, so that we can have like an idea of the different pathologies where they are overexpressing or they're failing, so that because we're all mostly in the clinical area, that would be of great interest for us.
Yeah, there is a, well, so as I said, connexin are almost on every cell of your body. So clearly they are connected with tons of different diseases. What is, well, we are more specific in the sense that we are targeting any channels that are leaking. So we can actually find something that works if the disease is due to the fact that the AMI channel is leaking. There are if the problem is done by the fact that AMI channel are opening, that can be also the case. So apart from genetic mutations that they have, there are genetic mutation for the skin and for the eye.
Autoimmune diseases also?
Sorry?
Some autoimmune diseases.
Autoimmune diseases, I'm not sure about that. There are diseases of heart and on the liver. So heart and liver can be targeted. Liver should be connexin 42. Okay. And there are many. Just not 100% sure.
Okay, thank you for the answer. I'm not sure the name of the panelist.
I would need to think about it.
I'm not sure the name of the panelist, but he's asking if you're aiming to expand your research subjects into something that could be potentially considered a bigger pathology of interest that could become like an innovation, and maybe you could patent one of these antibodies, similar to what Professor Lerner did before with some immune diseases, and he developed the Humira antibody. So are you trying to now aim all of this knowledge to potentially bigger or a bigger candidate of pathology?
Well, we are working mostly on parallels, so we keep working on this, and changing connexin can give the possibility to get on bigger pathologies or more frequent, let's say. It's an option, but yeah, we're still discussing about it.
I also remember one of the publications you did. You were assessing some antibodies with molecular dynamics for COVID-19, no?
We did a study of mostly it was like we studied the COVID-19 and different mutations of the COVID-19 and the ability of this mutant COVID. Well, we were especially looking at the spike protein, of course. So the spike protein and the different different mutation of the spike protein and how they interact with the ACE2 receptor. We did a work with that, and we also applied these mutations on to the interaction with one antibody and a couple of small molecules that were designed to block the spike protein.
Okay, nice. I will share that study with the students once we finish. Angel Noriega is asking if the software for MD simulations, do you need to have licenses to use them, or are there's some of these softwares that are completely free to use?
Yeah, there are some that are commercial and you need the license, and some that are free. Most of this work has been done with Gromax, and Gromax is free software, and it's quite fast. And another software that that I can suggest is OpenMM, and this software runs in Python.
Perfect, perfect. Okay. Juliana is now asking which area of research is the one that you've liked the most, and which one of your investigations or your research areas has been your favorite?
I didn't hear you, sorry.
Okay, which area of research is the one that you have liked the most, and which one of your investigations have you consider is your favorite one?
Well, as I said, working with the development of this antibody, I really like that. And also, as I said, I like to code, so I really enjoyed the programming of the old software for the in silico maturation of the antibody. I guess these two.
Perfect. Perfect, perfect. That's a very, very cool part of making mutations on an antibody to make it to have more high affinity. And our last question is, how long does it take approximately to develop the experimental part of one of these projects?
Oh, mostly depends on how lucky you are, but I guess it can take months on average, at least months.
If you're lucky, no?
I think the first part of the finding antibody and all the experiments before publishing the first paper, it was 1 year.
Okay. So yeah, usually the experiments can be everything can be perfectly smooth and go fast, or you can always have like small stones in the way that you have to manage to go around to develop and to succeed.
Absolutely.
Okay, Damiano. So we finished all of our questions from the students, and before we finish the talk again, let me thank you so much.
Thank you.
As for me personally, but also from part of the university, from Universidad Autónoma de Baja California, because this was an amazing talk for the students, and my idea was always to show the students that you can do science in the very frontiers of knowledge, and you can actually, if you're very dedicated, you can be able to work in very important labs, such as what you're doing with Francisco Sonta, and currently with Professor Michale Levitt. So let me just give some goodbye part for the students, and then we can close the talk, and then I'll just communicate with you through the WeChat, okay? So give me just one moment. Bueno, chicos, jóvenes, muchísimas gracias por asistir a la plática. Nuevamente, este ciclo de conferencias nosotros lo organizamos con mucho gusto para poderles brindar la posibilidad de conocer lo que se hace en la frontera de la ciencia, en laboratorios de investigadores importantes. En el caso del doctor Damiano Buratto, él tiene muchos proyectos. Si ven después su perfil van a encontrar que colabora con muchos laboratorios debido a que su habilidad para hacer dinámica molecular y otros modelajes de proteínas. Él puede diseñar anticuerpos de alta afinidad, él puede hacer modelajes de proteínas para relajar las proteínas para que se pueda determinar la estructura. Él de hecho también hace experimentos con los semicanales. Y bueno, espero que la plática les haya sido de su agrado. Vamos a tratar de continuar invitando investigadores de nuestra facultad o de otras facultades de la UABC, del exterior, tanto de otras universidades y del exterior del país, para continuar brindándoles un mejor panorama o un panorama más amplio de todo lo que se puede hacer. Si uno es dedicado, uno tiene interés, uno se puede enamorar de los proyectos y uno puede ir de la UABC a trabajar con un premio Nobel. ¿Sale? Entonces, por parte de la Universidad Autónoma de California, de la Facultad de Ciencias, Químicas e Ingeniería, de la Dirección, de las coordinaciones de posgrado, de nuestra facultad y de extensión y vinculación, les damos las gracias por haber atendido a la plática y les deseamos una excelente tarde. Bueno, mañana y tarde. Damiano, thank you very much for your talk. Again,
Thank you.
Amazing talk.
Thank you all.
And I'll contact you through the
All
weekend.
very interesting question also. Very nice.
Perfect. Okay. Entonces, we're gonna finish the closing the call, and I hope everybody has a great day. Cerramos entonces la llamada y que todos tengan una excelente día y tarde.
Bye.
Goodbye.
Recording stopped.